Hello and welcome to another video on genetics for school. Now in this particular video we'll start to learn about Mendel's law. Now in my previous video I have introduced you to Gregor John Mendel, his history, the usage of pea plant. Mendel, this guy, he was a friar, he was an Austrian monk, and he was interested uh, in inheritance patterns. So he, out of curiosity, started conducting certain experiments in the backyard of the church where he was basically working. Now he conducted an experiment on pea plants, and why he chose the pea plants is something that I have already dealt with in another video. And his experiments, he basically conducted his experiments for around 8 years. So that's a very long period of time. So these experiments were really time taking, tedious, but extremely well planned. And he chose pea plants for particular reasons. He raised a lot of them, collected a lot of data, which we will see soon. What kind of data did he collect, etc., etc. Now, he had grown like thousands of plants for 8 years, and then he came up with these different conclusions, which today we know as Mendel's laws. Now, what kind of an experiment did he conduct on pea plants? And those were hybridization experiments. And a hybridization experiment is nothing but transferring a pollen, which is a male gamete containing structure, from one plant to the basically stigma of another plant, which is nothing but the receptive part of the female flower of another plant of the same species. And then fertilization event would take place within the ovary, and you'll get seeds, and then you can start observing certain characteristics of the seed or the plants that will come out of these seeds. So basically such experiments are known as hybridization experiments. Now let's move to the next part. He conducted a couple of these hybridization experiments, and hybridization experiments can be of different types, right? Now, who conducts these hybridization experiments? Hybridization experiments are conducted by geneticists and also by breeders. Now, these are not necessarily hybridization experiments, but hybridization is carried out by geneticists or by plant breeders or animal breeders. Now, we've already dealt in some other video why geneticists do conduct these experiments. They conduct these experiments in order to find something, verify something, know something, whereas plant breeders or animal breeders simply hybridize in order to obtain a desirable breed, a newer breed with better characteristics, or to retain or preserve a certain breed, right? So that is their interest. So there are different types of hybridizations. When a breeder or a geneticist is only interested in one trait while conducting a particular hybridization experiment, if a breeder or a geneticist is only taking into account one observable trait, we say that the geneticist or the breeder is conducting a monohybrid cross. Now, let me just give you an example of a monohybrid cross. Now, for example, we have plants, and plants have wide variety of features. For example, the flower position is one trait, and then flower color is another trait, and then the arrangement of leaves will be another one, and the kind of seeds that they produce or the size of the fruit, etc., etc. These are all different traits. When a breeder or a geneticist is only interested while conducting an experiment, he's or she's only interested in one of the traits. For example, the breeder is only interested in color, and he's conducting a cross between, let's say, two plants for the improvement of color. Let us assume for a moment, for the improvement of color. Well, when the cross will be conducted, the outcome will affect all the characteristic of these two plants, but the breeder will ignore all the other characteristics, and he is only interested in studying the effect of this hybridization experiment or this crossing for one particular trait. Then we say that the geneticist or the breeder has conducted a monohybrid cross. If the breeder is interested in two observable traits, if he's observing two traits and during hybridization he's only observing what is the outcome of hybridization experiment on two traits, then we call it a dihybrid cross. Similarly, we can have a trihybrid cross or a tetrahybrid cross, but as the number of observable traits increase in an experiment, the experiment data also becomes complex. So anyways, let's not go there. Let's simply move on to what Mendel did. Now, he first of all conducted a monohybrid cross, and he chose pea plants. Now, why did he choose pea plants? Let's just do it again, because there are like 7 contrasting traits that he chose. There were other characteristics, but he only chose 7 contrasting traits. So these characteristics were the seed shape and the color of the seed, and the pod shape inside which you find the seeds, and then the pod color, right? And then we have the flower color, flower location, and plant size. And there were these contrasting features. For example, in a pea plant, you either found you find round seeds, and you can also find wrinkled seeds. Likewise, you can also find that the seeds are either yellow in color or they are green in color. Similarly, pod can be shaped as they can be inflated pods and constricted pods. We also call them the pinched pods. Well, you might have seen all of these features in pea. Also, the pod can either be green colored pod or they can be yellow colored. The flower of the pea plant can be purple or they can be white. Similarly, the location of the flower can either be axial, which we have dealt in the other video what is axial, and they can be terminal, or the plant can be tall or dwarf. So these were the 7 contrasting traits that Mendel chose for observation in his experiments. And the first thing that he did, he conducted monohybrid cross on all these 7 traits, which means by which we mean that he first of all he conducted a hybridization experiment only for, let's say, the seed shape. He was only observing the seed shape in one of the experiments. Then he conducted another similar experiment, taking into account seed color and then pod shape and then pod color. So this was a very elaborate experimental setup that he had conducted. Anyways, let's proceed further. So, but what made Mendel basically proceed in this direction? Why did he choose to conduct these experiments? Well, during his time, during the 19th century, there was a belief that inheritance, whatever characters we inherit from our parents, is basically a blend of the character from the father and from the mother. But Mendel observed, and it was a basic common observation, that the characteristics, yes, we do inherit characteristics from our parents and even from their parents, but the character is never a perfect blend of the traits that we acquire. For example, if somebody's father is, let's say, 7 feet tall and the mother is, let's say, 5 feet tall, the offspring will not be an average. It won't be, let's say, 12 by 2, wouldn't be 6 feet tall, right? So, Mendel was actually thinking that, well, whatever characteristics we inherit, they are not a perfect blend. They are usually not. They are mostly not a perfect blend. Yes, sometimes they are a blend, but sometimes the characteristics are altogether missing. So he was interested, he was basically curious to find out that what are the laws of inheritance? How are these characters that we inherit from our parents inherited? Do we inherit from one parent, or is it really a blend? Well, so during the 19th century, this was the belief that the inheritance was blended. So he started conducting his experiments on the pea plant, and then he conducted the monohybrid cross. And let's look at one of the monohybrid crosses here. And in the first experiment, I don't know whether this was the first one or the second one, but in one of the experiments, he chose round seeds, and he chose pure lines of round seeds. And he also chose pure lines for wrinkled seeds. Now, the round seeds, the plants, this particular trait of round seed is the dominating trait, and the wrinkled seed is a recessive trait. But he had no idea about this, right? That whether which is a dominating trait, which is a recessive trait, he had no idea about it. But he simply grew pure lines for plants in which the seeds always were round. And he also, first of all, developed a pure line for pea plants that had only wrinkled seeds, right? So just imagine how much of time this might have taken him. So after doing this, also, let me also introduce you to 2 more points, that during this time, the concept of genes was unknown to the human civilization. We had no idea about genes, we had no idea about DNA, nothing whatsoever. We probably knew about the nucleus. Well, there was something inside, but we didn't know what the role of the nucleus was. Also, there was no concept of the Punnett square. So how to analyze the data? With ease we do it today, but there was no idea of there was no concept of the Punnett square. So despite all of these limitations, he started his experiment and he concluded something really phenomenal, which we will explain side by side using the Punnett square in this video. And also, I hope you know what genes are, and that will become this information that what genes are will make it more easy. It will make us, put us in a very comfortable position to basically understand the outcomes of the experiment. Anyways, so let us see what he did. So he first of all chose these two pure lines. He chose a pure line for round seeds. He chose a pure line for wrinkled seeds. What do we mean by that? That this is homozygous dominant and this one is a homozygous recessive plant. And then he basically crossed these two plants. Well, which one is a male and which one was a female? He also conducted an experiment which is called a reverse cross, which you do not need to know now. And he figured out it doesn't matter which one is a male, which one is a female. The outcomes remain the same. So it just doesn't matter. So after crossing, so when these two plants are crossed, we know through the Punnett square, but let's not go there. What he found out that at the end of the experiment, he found that all the plants gave rise to seeds that were round. So this is the parent, these were the parents. So the parents, what was the genotype of the parents? Here is the genotype of the parents. He had no idea about the genotype at the moment because genes were not known, but they are known to us. So, but he used to call them factors later on. So let us just say that he took round seeds and he took the plants with wrinkled seeds, and then he made a cross. And after the cross, the seeds that he got, so after the cross, what happened? These plants will make some seeds. He collected those seeds. He put them in the soil. He put those seeds in the soil. When those seeds germinated and gave rise to a new pea plant, right? When it gave rise to a new pea plant, and when it gave, when the seeds were formed in this particular one, these seeds, they all were round, and the trait of wrinkled seeds had disappeared somewhere. So this is the first point I want you to remember. The parents were homozygous dominant, one of them homozygous recessive, the other one was round, the other was green. After crossing the first generation, we call it the first filial generation, or the first generation, the seeds were all round. Now Mendel was astonished. He was like, what the hell happened to the wrinkled seeds? Where did they go? Now, this was the first point of observation. Anyway, so what he did next was, he didn't stop there. What he did was he selfed these plants. Now, basically all of these plants were self-pollinated. And after self-pollination, he got the second generation. We call it the F2 generation. And after selfing, what he found was, he got two types of seeds. He found large number of round seeds and he found a certain number of wrinkled seeds. And the ratio of those seeds were 3:1, which we will come to in just a little while. So let's just go back and see. So this was the result that he was observing. Right, now let us use the Punnett square to basically decipher what was happening there. Let us just use the Punnett square to see what is happening around. So as the parents were homozygous dominant and homozygous recessive, I've made a Punnett square right here, and let us see what is happening. Now, I've told you it doesn't matter which parent is the male, which is the female, but I've just written it on the top. So the Punnett square is here. So if you look at the Punnett square, we see we have capital R coming from the homozygous dominant plant and small r coming from the homozygous recessive plant. And when we make a cross, we get four possible combinations, four possible outcomes. And what is this? This is a heterozygous dominant outcome. And that is why we get all the plants that are round, because the dominating allele R, capital R, is basically, it gives rise to round seeds. And the wrinkled trait is a recessive trait. It is dominated by capital R. So all the seeds that we get in the first generation are round. So what is the genotype here? The genotype in the first generation is capital R, small r, and the phenotype is round. And what is there any particular ratio? Well, all of them are 100%. So 100% genotype is capital R, small r, which is heterozygous dominant, and all the phenotype is 100% round. Now, after selfing, what will happen? And I've built a Punnett square somewhere here. Have a look at this particular Punnett square down here, and you can actually see I've done a Punnett square for selfing. So capital R, small r, capital R, small r. On selfing, what do we get? We get capital R, we get these combinations, right? Now, here is the interesting thing. Mendel had no idea about the phenotype, so what did he get? He gets round seeds and wrinkled seeds because there are like three. Let's look at this. So, first one, we know this. Mendel didn't know this. So, these are round seeds, capital R, capital R, capital R, small r will also give us round seeds because this is this is heterozygous dominant. And again, we get capital R, small r. So, 3 out of 4 outcomes were round seeds, and only 1 out of 4 seed was a wrinkled seed. And this is what exactly Mendel found, right? Now we have solved this, what happens in the second generation, right? And if you have to, but Mendel had no idea about finding out the, he couldn't find out about the genotypes because it was not known. So now Mendel also concluded a couple of things which we will discuss at the end. But Mendel didn't stop here. So he got 3:1 ratio of round to wrinkled seed. He said that out of all the seeds that I have, 3 parts are round seeds and 1 part are wrinkled seeds. Now he didn't stop there. What he did was he took all of these seeds. So he took, let me use a red marker and then proceed further. So let us say he took these wrinkled seeds, and what he did with the wrinkled seeds was this. He selfed the wrinkled seeds. So I'm writing number 3 here. Look at the third table down here. So he did this. He selfed the wrinkled seeds. Now, as wrinkled seeds were homozygous, what he figured out was that the result of selfing was 100% wrinkled seeds. And what did he conclude? He concluded that, well, he has basically obtained a pure line here. So he had obtained a pure line for wrinkled seeds. Similarly, he took some round seeds, and I'm only going to take this one at the moment. So he took 1 part of the round seeds, and he didn't know which one was which. So he took round seeds, he put them in the soil, and he figured out that, well, some of those round seeds were also pure lines, some of them. And then there were these round seeds that were remaining. And when he put them in the soil, he selfed them, when he selfed them, and this is the result of selfing them, he got a result which was similar to the cross of the F1 generation, to the selfing of the F1 generation. And now, if you look closely here, on the basis of this, he came to a ratio called 1:2:1, which means that this 3:1 ratio was the phenotypic ratio. This is called the phenotypic ratio. What does this mean? It means that there were 3 round seeds for 1 wrinkled seed in the entire lot of seeds that he had gotten in the F2 generation. But when he further analyzed it, he came to a certain conclusion. He said, well, there is something strange about 3:1. Basically, this 3:1 is the phenotypic ratio, but only out of all the 4 outcomes, 1 outcome was a pure line for the dominating trait. 1 part was a pure line for the recessive trait, and 1 line was 2 parts out of 4 were heterozygous, right? I hope this much is a little bit clear to you, and then let us proceed further. So this was basically the experiment that Mendel had created or performed, and these were the outcomes that we've just seen now. Now, let's just directly move on to the conclusion, and then we'll look at this particular table. So now, what did Mendel conclude after performing all of this particular experiment? So he said that, well, the idea of blending is wrong. There is nothing as blending that happens. Inheritance is not blended, it is particulate, or rather, it is carried by certain substances which he called factors. Today, we know these factors as genes. And why does he conclude that? How could he conclude that there is no blending? Let us just go up, and let's look at the parents. So, well, the parents were pure lines, right? One was a pure line for round seeds, the other parent was a pure line for wrinkled seed. If inheritance was blended, then the first generation, in the first generation, the offspring should have been a blend of round and wrinkled. They could have been less wrinkled. They could have been a little round and little wrinkled, but he found Nothing of that sort. He found only and only round seeds, only round seeds. And to his surprise, the wrinkled seeds were absolutely missing. Right now, that is a mystery that we'll come to next. So I hope you've understood the first point. Why could he conclude? So he concluded that inheritance is not blended, it is particulate, right? It is basically transferred by particles. Why did he say so is something that we'll look in just a moment. Now, the second conclusion that he made was there are certain factors that were responsible for the characteristics or traits. He said that there is something inside the plant, and he called that particular thing as factor. And he said that these things that are inside the plant are controlling the traits. Some are making some traits, some of these factors are controlling height, some of these factors are controlling the seed size or the color of the flowers, etc., etc. Today we know that these factors are known as genes. So let us just replace for a moment these factors by the word gene, and he basically concluded that genes were responsible for traits or characteristics in plants. He didn't stop there. He also said that there are always a pair of these genes present. One of them is a dominating partner and the other is a recessive one. Now, how could he conclude these 2 points is something is that what we'll look now. Now you see, let us look at this. So in the first generation, let me use a little eraser to rub off the mess that I've created here. Just a bit of it, the mess, right? Let me just, okay. So in the first generation, if you look closely, the parents are pure lines. One is round and one is wrinkled, but in the first generation, we get round seeds, correct? We get only round seeds. Wrinkled is missing. But all of a sudden, after selfing in the second generation, the wrinkled seeds reappear. Can you see here? Let me draw a circle in the fourth column of the Punnett square. Please have a look here. And here we see in the second generation, the wrinkled seeds with the genotype smaller had reappeared. So Mendel concluded that for some reason which was not known to him, he said, in the first generation, even though this trait wrinkled, see, how could wrinkled seeds appear again? From the first generation, how could they appear? So he could only rationally conclude that something, the trait for the wrinkled seed was suppressed by some factor, and he concluded, well, there are a pair of genes or a pair of these factors, one of them dominated the other one and hence it was not expressed, but the other factor, the recessive factor, expresses, it doesn't go missing, it is hidden, it is the recessive, and when it gets its own opportunity, it expresses itself. And when does a recessive character express itself? It only expresses itself when it is in the homozygous condition, when both the alleles are recessive. So I hope now that you have understood the second point and how Mendel was able to conclude that. What were the bases? The second point was that genes were responsible for traits. There is always a pair of genes present for one character or one trait in a particular organism, and one of this particular factor or gene is the dominating one and the other is the recessive one. Thus, a pair of gene is responsible for or basically controls one trait. Now comes the third and the most important part, which is the reason why this particular outcome is also known as the Mendel's first law or the law of segregation. Now, Mendel concluded at the end of the experiment that the gene pair, you see, we are talking about this gene pair that we have just discovered. He said that there is a pair of gene present controlling one trait. He said that this pair of gene is usually found in all the cells of the organism, but he said that when gametes are being formed, both these gene pairs or both the partners of this gene pair, they segregate, they separate from each other when during gamete formation or gametogenesis, and each gamete receives one of this gene. Each gamete receives one of this gene from this gene pair. Let's just have a look up here. So what Mendel wanted to say is this, that for example, let's just look at this particular variant. He said that there is a pair of gene present in the organism, and this pair, basically both the genes separate from each other, they segregate from each other when during gamete formation, and each gamete will then receive one of these factors. So each gene has one factor from the allelic pair, and this is what is today known as the law of segregation. And what is the statement for law of segregation is this: traits or characteristics are segregated or separated into gametes, and each gamete contains only one allele from their pair. So there are so many characteristics in all of us, all the diploid organisms. So for example, let us just stick to pea plant. So a pea plant has a characteristic of height, flower color, flower position, seed color, seed size, etc., etc. And each of these characteristics is controlled by a pair of genes. Let's say AA, let's say BB, let's say CC, let's say DD, and let's say EE. And what else? He says that when gametes are being formed, so let's say these are gametes are being formed, he said that each gamete will only receive one character from each parent. So you see, okay, let me just make just two gametes. So let's say there are, let me just make two gametes. So let's say this is one gamete, and there can be like hundreds and thousands of other gametes, but according to Mendel, each gamete will receive only one of these allelic partners at random. So, but it will only be one. Both the gene pairs will not enter the single gamete. And then these gametes, eventually, you know what happens in fertilization. I hope the law of segregation, as discovered by Mendel through his experiments, through his exhaustive experiments, is clear to you. Now, before I come to an end, I want to present to you a certain data table, looking at which you can figure out how tedious his experiment was and what a brilliant genius this person was. So he conducted his experiment on all the 7 characteristics. He conducted these monohybrid crosses on all the second experiments, and let me just magnify it a bit so that you can see it a little closely. So for the first experiment, the original data that is available to us today, that we know that he performed, he actually, from one of his crosses for round and wrinkled seeds, in the first generation he found all round seeds, as we've seen. And in the F2 generation, what is the ratio there? It's 3:1. It's the phenotypic ratio is 3:1. The genotypic ratio is 1:2:1. Now in the second generation, he basically found 5,474 round seeds, and he collected 1,850 wrinkled seeds. Now, if you simplify these two, you will get a ratio of 2.96:1. This is not exactly 3:1, but this is basically rounded off to 3:1. In the second experiment that he performed, which was a monohybrid cross for yellow and green seed color, first generation certainly will belong to the dominating character. And in the second generation, he obtained 6,022 yellow seeds and 2,001 green seeds. So he basically had counted all of these seeds. Every single seed, there would have been loss of seeds and blah, blah, blah. And there he got a ratio of 3.01:1. Now, if you look, forget about all the rest, but if you look at all of these 7 traits and the number of seeds that he obtained, or the flowers that he saw, or the seed pods that he saw, you see, look at this ratio of F2, 3.15, 2.95, 2.82, 3.14, 2.84 is to 1, all of them. So these are not perfect 3:1 ratios, but close to 3:1 ratios. I hope that you have understood the law of segregation, what Mendel concluded, what were the 3 major conclusions that he made, and what is law of segregation in genetics. And rightly so, Mendel is also known as the father of genetics. I will see you in another video where we will talk about Mendel's second law, which is known as law of independent assortment. Thank you very much.